Okay, this is chapter 12 in muscle physiology. So we'll get started with the outline. The first part of this outline deals with the contraction of skeletal muscle and the structure of skeletal muscle. So the structure and contraction is probably a little bit of review from anatomy, but I'm going to go through it to give you that review. So this is the structure of skeletal muscle. Remember that we have skeletal muscle cells are called fibers. Okay, so they're long, they're unbranched, they have many nuclei because they were formed with the fusion of these cells called myoblasts as an embryo. So multinucleated, it's striated, it has a striping pattern on it due to the overlapping actin and myosin filaments in our structures called sarcomeres. If we look inside of a muscle fiber, we can see that it's filled with these protein filaments, and that's going to be your actin and myosin. So actin and myosin, remember, actin is thin, it's a thin filament, and myosin is thick, and the way that they overlap creates a sarcomere. The train of sarcomeres, right, the sarcomeres, right, one against another, create a structure called myofibril. So the myofibril is going to be the contractile unit of the muscle cell, and it's created from these, different, or the sarcomere down the myofibril. The sarcolemma is what we call the plasma membrane of the muscle fiber. So remember that sarco is used a lot. So sarcolemma, sarcoplasmic reticulum, and then the sarcoplasm would be the cytoplasm inside. Now, it's arranged in this manner where we have many fibers grouped together within a fascicle. So a fascicle is an organized unit of muscle fibers. The connective tissue that does this is called perimysium. Perimysium. So between the muscle cells, the muscle fibers themselves, and also inside the fascicle, remember we had a connective tissue called endomysium. That's the innermost connective tissue. So endomysium is seen here pointing inside of a fascicle. Once that endomysium comes outside the fascicle and creates that fascicle, then it's called the perimysium. So the perimysium is the connective tissue that surrounds all the fascicles, fills in the spaces around the fascicles, and creates those fascicles. Then when the connective tissue comes up to the surface of the muscle, it's called epimysium. Okay, so our skeletal muscles are organized in this fashion, and fascicles can contain anywhere from hundreds to thousands of muscle fibers. And so we can also see, should be a review as well, that the epimysium, the connective tissue here, can thicken to form a tendon, and our tendons are going to attach our muscles to bones. So remember, the definition of a tendon is a muscle to bone attachment, and the word ligament means bone-to-bone attachment. All right, so moving on to the inner part of the microanatomy of a muscle fiber, coming back into our myofibrils, just to take a look at our thin and thick filaments. We'll take a look at a sarcomere in just a second. Notice that we have many mitochondria within the muscle cell because muscles are always using ATP. But when we see how a skeletal muscle gets innervated, here is our motor neuron. So our voluntary skeletal muscle is innervated by motor neurons. Motor neurons are going to come from the spinal cord. If you remember from anatomy, the body of the motor neuron is in the anterior horn of the spinal cord. And it's going to come out and innervate your muscle fibers. So in this sort of internal view, we want to remind ourselves that we have transverse tubules, these T-tubules. So the sarcolemma, right, the plasma membrane of the muscle fiber itself, contains these invaginations, these sort of tubes that travel down into the muscle fiber. So here's another image of a T-tubule and how the sarcolemma dips down to become the T-tubule. The transverse tubule here is going to lie in between two sides of the sarcoplasmic reticulum. The sarcoplasmic reticulum has these special areas called lateral sacs. These lateral sacs are going to store calcium. Okay, and the transverse tubule is going to lie right in between two lateral sacs. So when the impulse from the motor neuron comes down into the transverse tubules, that impulse can be relayed to both of our lateral sacs from that transverse tubule. All right, so let's take a look at our sarcomere. Oh, and by the way, so just moving through your outline, this is called a triad, where you have the transverse tubule and 2 lateral sacs on either side. And this is our sarcomere. So review your sarcomere structure. Remember that a sarcomere is defined by 2 Z lines. So our dark Z lines on either side here. And what's attached to the Z line will be the actin or the thin filaments. So you see in the picture here, we have the Z line in purple, then we have the purple kind of extending out, that's going to be actin. And then your myosin is kind of this golden colored filament. And the patterns that we have here, refresh your memory, right? So we have our ZIAH. So our Z lines, I always like to do it this way. When you walk in, we're going to move towards the M line, that midline. So the Z line, the first area you come to is the I band, okay? So the I band is going to be where you have only actin, okay, in the relaxed sarcomere. So here's our I band. We only have actin. Then we have the A band, and the A band is going to have an overlap of actin and myosin. So this is where we have actin and myosin together. So it's going to be darker in color because the I band only has the thin filament. Remember, actin is thin. And so if you took a picture of it, it's just not going to be as dark because you don't have as many protein, like as much protein there because it's a thin, only actin. But when you get to an overlap where you have actin and myosin, then it's going to be a lot denser and darker in color. So the I band is going to be dark. And then the H zone in the center is going to be where actin is no longer present, but you still have myosin. So the H zone is only myosin. But so it's going to be lighter in color, and then you have that M line in the middle. So you want to remember what happens to a sarcomere when it contracts. What changes in these different structures of the sarcomere. So I have these written down a little bit further in the lecture. If you look at Roman numeral 2, the sliding filament model, actin slides over myosin in muscle contraction, and when the sarcomere, within that sarcomere, what happens is, all right, so here's a bigger picture, right? So we can take a look at this. So what happens is our sarcomere contracts and our Z lines are pulled in towards each other, towards the M line. So we can see that what's happening is our I band, where we're going to have actin, is going to start to get smaller because we're starting to overlap more and more that region on the A band where we have an overlap of myosin and actin. So actin's getting pulled in towards each other, increasing that overlap and decreasing where you only have actin. So the I band, right? The I band here is going to get a lot shorter. It's going to be minimized compared to the I band over here. The A band stays the same because this is basically defined by myosin. So the A band doesn't change when the muscle contracts, but the H zone does get smaller as well because remember the H zone is where you have only myosin. But if you're pulling actin towards the midline, you're going to have more and more overlap where you just had myosin in the H zone, but you're going to have more and more overlap. So that H zone is going to get smaller because you're going to start to pull actin in and start to overlap with myosin. So if you look at your notes, it says the A band stays the same, I band shortens, the H zone shortens, and this whole sarcomere shortens, right? So we can look at this picture too, right? So the Z bands come in towards each other. It's not as distinct here, but the I band is going to get shorter, H zone gets shorter, right? We can see that where we just had myosin gets very small here. And the whole sarcomere starts to contract. All right, so let's move back up and talk about our filaments. All right, so this is the thin filament. This is actin. So actin's made of these globular proteins called G-actin proteins, and G literally means globular. So it's a roundish, blobby protein, which is what globular means. You just put it together in a filament, and there's a really important part on actin called the myosin binding site. So the myosin binding site is always pictured as like, they look like olives to me. It's the pit in an olive. And on top of actin, you have two more proteins. One is called tropomyosin. So tropomyosin is a really thin filament, this purple string that lies on top of the myosin binding site. So it covers it up when the muscle's relaxed. Then we have another protein called the troponin complex, which is really 3 proteins in a row. And that troponin is going to help bind calcium, and it lies on top of tropomyosin. So troponin lies on top of tropomyosin. And when calcium binds to troponin, so it has a calcium binding site, it's going to shift the tropomyosin out of the way so that it exposes those myosin binding sites that actin has. Okay, so that's basically in number 2 in your outline, where we have 2 regulatory proteins on top of actin, tropomyosin and troponin. So troponin, I just say that it has a calcium binding site, and it helps to expose that myosin binding site. Here's myosin. So myosin looks like a golf club that are kind of intertwined around each other. And myosin has the active part of myosin is called the head. And the head has 2 important parts to it. It has an actin binding site, because we know it needs to bind to actin, and it also has an ATP ACE site. So ATP ACE is an enzymatic site that breaks down ATP. We can see that the way the whole filament is put together, you have a lot of these myosin filaments bound together, and the heads are going to be part of the zone that binds to actin and is actively taking part in the contraction. But in the middle, there's a bare zone, and this bare zone is just where we don't have any myosin heads, so it's not going to be active in contraction. All right, and then we have another protein called titin, and I don't think, oh, here's a picture of it. So we have this titin, and it's very elastic. It can stretch up to 3 times its length, and it's basically associated with myosin. So you can see how it's kind of touching and tagging myosin on both sides here. And it looks like a spring, right? So as your muscle is stretched, right? So the muscle is stretched, the titin is also stretched, but it kind of resists that stretch. And when the stretch is over, it pulls the sarcomere to its original length. So it also, so there's that one job, and there's also the job of just anchoring the myosin in place. So titin is another protein filament that's in your sarcomere. All right, so let's take a look at the sliding filament theory. Oh, so a quick question for you. What's the name for the dark striation observed under the microscope that is due to the overlap of thick and thin filaments? Okay, where we have an overlap, that's called the A band. Okay, so let's talk about the crossbridge cycle. So this should be a little bit of review as well from anatomy. So the way that your muscles contract is that the myosin heads are going to grab onto actin, and they're going to pull actin towards the midline. So both sides of your sarcomere are going to get pulled. The actin filaments will slide over the myosin filaments until the actin filaments meet in the middle. So let's start at number 1 here, right? So there's a binding of myosin to actin. And remember that this is only possible, I'm just going to reminder here, that's only possible when there's calcium present. So this little green ball right there, that's calcium. And there's another green one. So calcium is responsible for binding to that troponin, and the troponin is going to move tropomyosin out of the way. So we can see that we have a hydrolyzed ATP, so we have a breakdown. And remember how phosphate, when something is phosphorylated, it's going to be sort of unstable and more likely to react. So it's phosphorylated here. We're going to kick off the phosphorylation, and when we do that, we're going to take this high energy form of myosin. So just moving back a second, this is a high energy form. So when myosin's head is cocked back, into sort of this position that you see here. It's kind of like if you had like, I don't know, one of those guns that you have to sort of load by, cocking back a spring, and then you shoot the BB gun or whatever it is, you let that spring go. So this, when you cock back this spring, right, it has a lot of potential energy there. So that's what this is basically doing. So you have this myosin head in a high energy position, and when you release that phosphate group, it's going to pull, that's going to release that high energy position, and it's allowed to move towards a different conformation into a lower energy form. So you can see that the myosin head, it's attached to actin, and it's going to push the actin in this position because it's going to change conformation, and this is towards that midline. So this is called a power stroke. The movement of the myosin head from this cocked back position into another conformation is going to be that power stroke. So we have a power stroke occurring. So actin gets pulled towards the middle of the sarcomere. Now the rest of that ADP is going to get released, and now we are in a rigor state, or a low energy form. So myosin heads that have just done a power stroke, right? They've used that energy in the ATP to do the power stroke. They've done this power stroke. So they're in a low energy form. It's still attached to actin, okay, but it's just kind of not going to be able to do another power stroke until we need to add an ATP. So a new ATP binds to the myosin head, and it's going to bind to that ATP ACE region. When this happens, the myosin is going to detach from actin, okay? So when it detaches from actin, the ATP is going to get hydrolyzed. So we're going to break down ATP. So the ATP binds, that ATP binding detaches the myosin head from actin, and then it uses that power to go back into the high energy form. So it breaks down the ATP, and now we're cocked back into this high energy form. And that high energy form actually has a higher affinity to actin than the low energy form. So it's going to bind to the actin, and then the process starts over again. So this is called the crossbridge cycle, and this is going to go again and again and again and again until basically, and that's what causes muscle contraction. So we're contracting the sarcomere and getting the whole muscle to contract. Let's talk about rigor mortis for a second. So if we look at our outline and we look at number 2, the rigor state. So we see this, or I've also heard this pronounced rigor. When a person dies, there is a period of time that their muscles become very, very stiff, right? That's where the word stiff comes from for a dead person. And this is because their muscles will all go through a state called rigor or rigor mortis. And what's happening is that when you die, the sarcoplasmic reticulum in your muscle cells need to work to pull calcium into it and to hold calcium and store it. But when you're dead, that mechanism no longer functions. So all the calcium kind of spills out of your sarcoplasmic reticulum. And when the calcium spills out of your sarcoplasmic reticulum, it binds to that troponin and it moves the tropomyosin out of the way. So what little ATP you have left is going to, remember you just need one, it's going to pull the myosin head into this, you're going to do a power stroke. And then that power stroke, you're going to be in this low energy position in a, it's going to be bound to actin. And if there's no more ATP, right, because you're dead, so there's no more ATP coming, you're going to get stuck in this position. Okay, so you can't do another cycle. You can't let go of actin to cock back and do another power stroke. You're stuck to actin in this low energy form. And again, because you're not breathing, because you're not making ATP and all your ATP stores are gone, you're stuck in this position, and that's rigor mortis. So your muscles are in a contracted state, right, in this low energy contracted state, until enough time has passed that you're literally the proteins just start to fall apart. And that's when you lose rigor and you become floppy again. But there is this one state. And this is due to the binding of the myosin heads to actin, doing a power stroke, but then kind of just getting stuck because we're using any ATP to let go, to unbind, right? Because we need ATP to unbind and then to cock back for another power stroke. So I think we kind of hit the crossbridge cycle there. And then let's just look at the whole story about how muscles are contracted. So you're going to get an impulse from your motor neurons, right? And the neurotransmitter here is going to be acetylcholine. So acetylcholine is going to be released into the cleft. And there's some terms here if you're not familiar. So the region of the sarcolemma that is going to be in contact with the synaptic terminal or the axon terminal is called the motor end plate. So the motor end plate is just a specialized area that's going to have a lot of acetylcholine receptors. It's going to be folded in because that's going to increase the surface area. You can fit more receptors into an area that has folds. If it were just flat, it just doesn't have that much surface area, so it's folded. Has a lot of acetylcholine receptors. So when acetylcholine is in the cleft, it's going to create an impulse or an action potential on the side of the sarcolemma, right? So if we're reading the slide here, right, the binding of acetylcholine is going to elicit an end plate potential, which then triggers an action potential in the cell. So our action potential is going to propagate down the sarcolemma and down the T-tubules, right? So the T-tubules are traveling. Remember, on either side of the T-tubules, we have those lateral sacs, which contain calcium. And the impulse is going to trigger the release of calcium from the sarcoplasmic reticulum. So the calcium's release is going to flood the internal cytoplasm of the muscle fiber with calcium, and then the calcium will bind to the troponin, and it's going to expose the myosin binding sites on actin so that myosin can bind and continue with our crossbridge cycle. So as long as there's calcium present, right, because we need calcium for muscle contraction, and as long as there's ATP present, we're going to continue to contract our muscles. Okay, so 5, 6. So calcium is actively transported back into the lumen of the SR following the action potential. So if the action potential stops, then the calcium is, as soon as the calcium is released, the sarcoplasmic reticulum continuously works to take it back in, right? So when your signal to relax the muscles stops, the calcium is already being transported back into the sarcoplasmic reticulum, so you clear the calcium within the cytoplasm so that you can get muscle relaxation. So troponin, so when calcium is transported back into the sarcoplasmic reticulum, then we have the tropomyosin moving back to its original position to block the myosin binding sites because the calcium is leaving the cytoplasm. Okay, so this is how it's called excitation contraction coupling. So we're exciting the sarcolemma with our motor neuron, and then we get contraction happening at the same time. All right. So this is twitch. So I think that covers, one more thing. Okay, I don't have a picture of it, but let's talk about, the receptors here. So muscle contraction, number 3. If you look at letter C in your outline under number 3, there's voltage-gated DHP receptors on the transverse tubules. They are linked with calcium channel receptors in the lateral sacs of the SR. So I'm just going to increase this slightly. And so as the action potential comes down the transverse tubules, there's receptors on the transverse tubule here. Okay. And these are your DHP receptors. Actually, they're not receptors. Well, yeah, they're receptors. So they're going to be linked. So on the sarcoplasmic reticulum side, there's another receptor called this ryanodine. So there's another one. I'll just make that uncolored in. So there's this ryanodine receptor here. Okay, and so they're bound together. And what happens is the DHP is voltage gated. So this one responds to voltage. So as the action potential comes down, this guy opens due to the voltage. it's going to have a conformational change, right? The voltage will cause an opening. It's going to change the shape of this protein. And that conformational change actually creates that change in the ryanodine receptor. So the action potential causes a conformational change in the DHP receptor, which then causes a conformational change in the ryanodine receptor. And that's going to cause the calcium to be released from the sarcoplasmic reticulum. Okay, so I hope that's clear. And that's it. So that's the end of our muscle contraction. And for a lot of you, this is review, maybe with a few more little details, especially with the DHP and ryanodine working together. But most of you guys have probably had that before. So our next topic is something called twitch. So the mechanics of skeletal muscle contraction, when your muscles contract, it's called twitch. So twitch is, if we only have one single action potential, what does the muscle do, right? What does the muscle cell do? So twitch is that response to just one action potential coming into the muscle. And it turns out that when you do have an action potential coming to a muscle fiber, it contracts and it has these distinct phases. So it has a latent period, it has a contraction phase, and then it has a relaxation phase. So there's 3 phases, right, to twitch. So just to talk about letter B, twitch can be defined for a muscle fiber or from a motor unit or even at the whole muscle level. But we're just going to talk about it right now in terms of just one fiber. So these are the phases. So the latent period is basically we have nothing going on, right? So we have the stimulus, we have the electrical impulse or action potential to begin contraction, but there's a lag of time, a very small lag of time. And that is because we need to wait a few milliseconds just to get that calcium to diffuse into the sarcoplasmic reticulum, from the sarcoplasmic reticulum into the cytoplasm, or I should say sarcoplasm. And it has to bind to the troponin, and the tropomyosin needs to move out of the way, and the myosin heads need to attach, and then you can get contraction. So that just takes a little bit of time. The contraction phase is we have crossbridge cycling, right? We have the power strokes. As long as there's calcium and as long as there's ATP in the environment, we can have the myosin head cycling or crossbridge cycling. The relaxation phase is going to be the phase where it's going to drop back to 0. So calcium is being actively taken back up by the sarcoplasmic reticulum, and we're going to start to relax and the muscle. And you can see it's a gradual phase. So the relaxation phase is gradual, and it's actually the longest phase out of the muscle twitch. All right. So there are a couple kinds of contractions that we see in muscles, and they are called, or twitch, we can just use the word twitch. There's isometric twitch and there's isotonic twitch. So I like to think of it in terms of exercises. So it corresponds to the whole, like at this point I'm talking about the whole muscle, but you can also think of it as one cell. So an isometric twitch, iso meaning the same and metric meaning the length, you're not changing the length of your muscle. You're not shortening it or contracting the muscle. You're not lengthening it, but you are creating tension. So the muscle's working. You have tension in the muscle, but it's just not changing length. So if that's hard to understand, you can just think about being in a plank position, right? So if you're in a plank position, you know your muscles are working. They're holding your body up. Both, if we just talk about your arms, right, both your biceps and your triceps are contracted, but there's no motion at the elbow. Your elbow's not moving, and the muscles are not changing length. So that is isometric contraction. Isometric contractions are all or nothing. They are reproducible, so they're basically the same kind of contraction over and over again. And and that's all it is. And basically that first letter A says, the muscle creates tension but maintains the same length, and it does not overcome a loading force. So what that means is if you're overcoming a loading force, that means you're basically picking something up, because a loading force is something that's heavy. So like a dumbbell or even just a coffee cup or a pencil, whatever you're moving is a loading force. So if you're not moving anything, right? If you're not actually changing position of your joints, imagine holding, a cell phone in your palm of the hand, but you're not in your arms extended, but you're not moving anything. So you're kind of holding your phone still. Your muscles are not moving. The joints are not moving. So you're not overcoming the load of your phone. If you were to pick up the phone, that would be overcoming the load. Your muscles are, the force and tension on your muscles is going to be greater than the load, so you can move the load. But in this example, the tension on the muscles is less than the load. You're actually not moving anything. If you look at isotonic twitches is when you're actually moving the joint. So if you look at number 4, letter A, muscles generate a constant force just greater than any forces opposing it. Forces opposing the muscle are called a load. So you are moving the object because the muscles are creating a tension that overcomes the load. So your tension of the muscle is greater than the load. So you're moving the weight. Now, you can move the weight a couple ways, okay? So what happens is you go through the full range of motion. So isotonic exercises involves putting a constant amount of weight or tension on your muscles while moving it, and you're moving your joints through a full range of motion. So I have an example, bench pressing. So if you're looking at the types of isotonic, you can have a concentric contraction where the muscle gets shorter. You can also have an eccentric contraction where the muscle actually gets longer. Okay, so but the idea is that you're actually moving the load, right? So for the example in your outline where you have the biceps brachii and you're doing a bicep curl, the bicep is going to get shorter as you're curling up, as you're flexing your elbow. But then if you're uncurling, I guess, I don't know if that's the right word, but if you're lowering the dumbbell, your biceps are still working. Imagine lowering it very slowly. It's still working it's contracted, but it's actually lengthening as it's moving, as it creates tension and as you're lowering your elbow or extending your elbow. So those are the two kinds of contraction. So concentric contraction was the muscle gets shorter. Eccentric contraction is where the muscle gets longer. Both of them, though, you are creating tension. There's tension in the muscle. And so I just put up a little, so you don't have to be doing like exercise to, work your muscles in these ways. Just having coffee with your friends, right? If you're going to pick up the coffee cup, then the tension in your muscle is greater than the load, right? So what kind of contraction is this? It's going to be isotonic contraction because you're moving the object. Your muscles are overcoming the load. So you're moving the object, you're, moving your joint. And so this is going to be a concentric contraction because if you are going to, just looking at the biceps here, right? So you're going to move that coffee cup up to this position to your mouth, and your biceps are going to contract. And so that's going to be a concentric contraction for your biceps. If you want to hold the coffee cup in one position, then you make the tension of the muscle equal to the load of the cup. So if you're going to hold the coffee cup in space, then your muscles are tensed. They have tension, but you're not moving anything, right? You're not overcoming the load. You're just holding the load still. So that's going to be an isometric contraction. Then let's say you're putting it back down. So if you want to go ahead and put your coffee cup back down, and we're looking at your biceps brachii, then we know that we have a motion, so it's isotonic contraction, but you're lengthening that biceps brachii, so it's an eccentric contraction. So even, you're constantly moving through these different kinds of contraction throughout the day. You don't have to be doing a physical exercise to do this. Measuring this in the lab, just real quick. So if you wanted to measure different kinds of contraction in the lab, this is for isometric contractions. So isometric contractions, your muscle is not changing length, right? So what they do is they anchor the muscle to two ends, and then they add a stimulator. So you zap the muscle with, it's kind of like creating an impulse, and then you'll have a reading, right? And so you should see the same kind of twitch that we have here. So we have a lag, we have the contraction, and we have relaxation. When you have isotonic contraction, isotonic contractions, the muscle will get shorter. So what they do is they basically anchor just one side of the muscle, and then they have a load on a freely moving. So this load can go up because you can see the difference here, because the muscle is going to get shorter and it's going to move the load or overcome the load. So one end of the muscle is free, it's attached to the weight, so it can shorten when it contracts. And then when you calculate or create the sort of twitch graph in this case, you always get this characteristic plateau. So what's interesting is you have a plateau that appears. And here is where the muscle's getting shorter. So you have your lag phase, and then you have a tension phase, but this is where you're just ramping up the muscle contraction and you haven't yet shortened the muscle. You're just overcoming the load. So you're overcoming the load. It takes a little bit of time to ramp up all the motor units you need to overcome the load. You're not shortening quite yet, but then there is this plateau where you are shortening. So you can see that in this graph here where you have the shortening doesn't begin until you hit that plateau. So it corresponds with the plateau. And then you hit that relaxation phase, right? So there's a relaxation phase, and you don't have shortening anymore, you're lengthening. Okay, so factors affecting the force generated by individual muscle fibers. So what's going to affect how much force one of your fibers can generate? So there are these three listed here: the frequency of stimulation, and that should look familiar, right? Because we know that the nervous system can stimulate any cell that it's talking to. It can send slow action potentials or very quick action potentials. So the frequency of stimulation is important. The diameter has a role, and the change in fiber length has a role. So let's look at the stimulation first. So there's a couple topics under the frequency of stimulation, or these phenomena. So one is called TREP, and this is where action potentials come so closely. Well, let me start off with, so here's the stimuli, right? These are the action potentials. So when you have an action potential that occurs, this is the tension, right? This is how much force that the muscle is creating with that initial stimulus. So notice with the first stimulus, our peak is low. With another action potential, another stimulus, the peak increases. With a third one, it increases even more. So with repeated stimulations, you can actually get an increase in tension until you reach the plateau. Okay, until you reach basically a maximum tension that can be created with these action potentials coming in. So they don't really understand why is it that with more action potentials, your muscle fiber can actually create more tension. And one of the things that was mentioned in the book was that, well, maybe you're just warming up. So the sarcomere at some level is more able to connect those myosin heads to actin. Maybe this first one, you don't have a whole lot of crossbridge formation, and as you warm up the muscle, you can actually get more and more crossbridge formation and get a more tension on the muscle. But regardless, this is a phenomenon that happens. So with more action potentials that come, you can actually get more tension created. Okay, so frequency of action potentials can give you more tension. The second one is summation. So when action potentials come in really quickly, the twitch can overlap and they can sum. So let's take a look at this, right? So action potentials are actually really fast. So the message coming in from your nervous system lasts a very small amount of time, anywhere from 2 to 5 milliseconds. That signal then causes your muscle fiber to contract, but the contraction, the whole sort of twitch cycle, right? The lag, the contraction, the relaxation, that takes a lot longer. It takes 10 to 200 milliseconds. So we can see that the overlap here, the pink being the action potential and this being your twitch. You can see how the twitch takes a really long time. So how many action potentials can you throw in there while the muscle is actually doing its twitch? A lot, right? So we can see in this picture here, we have our first action potential, we have our twitch. But if you send more action potentials, then you can sum up those action potentials. So you can actually build up, you can build tension. So if you wanted an analogy of this or an example of this would be like picking up something light. Your nervous system will say, hey, to pick up that pencil, we don't need a whole lot of action potentials, right? Because maybe this amount of twitch or maybe just a few action potentials spaced this far apart is enough to pick up your pencil or enough to pick up your phone. But if I wanted to pick up, if I'm working out, I want to pick up a 10-pound weight or grocery bags, right? A heavier grocery bag, then your nervous system is going to send more frequency of action potentials. And with that, you can sum up how much tension is created by your muscle fiber. So you can create more and more tension until when, right? Until you reach the peak tension. Your muscle physically cannot generate any more tension. You've maxed it out. It's as contracted, as much tension as your muscle can possibly create, and that's called tetanus. So tetanus is the plateau. It's the peak summation. It's where literally, molecularly, all your myosin heads have bound to the actin for maximum force. And any more action potential is not going to increase this force because you literally just maxed out how much your entire muscle force, right? All your myosin heads binding to actin. So that's summation. And then we talked about tetanus too, right? So tetanus is basically the peak of summation. All right, so those are some factors with how much force is generated by muscle fibers. Let's look at something else. Let's look at the diameter. Before we look at diameter, well, actually, I don't have a picture on diameter, but let's take a look at this. So this is just another way of looking at the topics we talked about. Here's one twitch. We have one stimulus, one twitch. This is the definition of twitch, right? It's what happens to the muscle fiber when you just have one action potential. Then we have summation. So if we send more action potentials, we can actually sum up and send more action potentials, but also closer together. We can sum up the amount of tension that can be put on the muscle, right? And if we have more and more frequent action potentials, we can achieve tetanus. Okay, so we don't need to worry about number 3, but complete tetanus is where you have maximum contraction. All right. So as for diameter, I don't have a picture of it, but the larger the diameter of the muscle fiber itself, so the cell, the bigger and thicker it is, the more force it can generate. Because imagine you just have like a really large protein-packed cell, right? If all of those proteins contract, if all those myofibrils contract, you just have more contraction power to contract more forcefully. If you have a thinner muscle fiber, then you don't have as many myofibrils inside, and when it contracts, you just can't generate as much tug or as much tension or force, right? So the size of the muscle fibers matter. And that's what happens when you get in shape or you build muscle mass, is that your muscle cells actually get larger in diameter. All right, and then there's the changes in fiber length. So this is just, it looks complicated, it does not have to be complicated. When your muscle fibers, when your sarcomeres are at its sort of resting state, that's when you can generate the most force. And they created this graph to basically tell you can't, so this is your tension. So when you're at 0 here, there's 0 tension. The muscle just cannot create any more tension. I like to start with C. So starting with this guy here, if your sarcomere is completely stretched out, so you're stretching your muscle, and you can see that the sarcomeres are able to be stretched, right? We have stretched it so far that there's completely no overlap. So myosin is not overlapping with actin. Can you actually get muscle contraction if there's no overlap, right? Absolutely not. The myosin heads can't bind to actin and you can't get any contraction. So there's no way to form a crossbridge. There's no contraction, no tension. As you move towards letter B, you can see the picture of the sarcomere is starting to overlap. when you increase the overlap, now you have areas for the myosin heads to grab onto, and you can have some tension being built, right? You can pull the sarcomere a little bit closer. So from C to B, a muscle can generate greater and greater force as the overlapping area becomes greater and greater. Until when? Until the optimum length, right? So there's an optimum length, which is in this green area at the peak here. So the optimum length of the sarcomere is basically the resting length of the sarcomere. And you have an optimum overlap of myosin. So the bare region is not overlapped because the bare region doesn't have myosin heads. It doesn't take part in contraction anyway, but you have full overlap where you have those myosin heads in actin. All right, so it's then the opposite. So let's now look at when we get closer and closer. So if we're going to start to overlap even more our actin and myosin and pull it in, you're going to get less and less area to form a contraction, right? So your I bands are getting short already. When you have contraction forming, there's not any further to go. You can't pull those Z lines any closer because they're already at the maximum overlap, at least in this picture, right? You're at the maximum overlap. How can you generate more force if there's nowhere to go, if you can't shorten anymore? So that's why this is not ideal as well. You can't generate force if you're going, or tension, if your sarcomeres are completely overlapped. So it's just kind of a curve to sort of illustrate what you probably already intuitively know, is that you have to have this overlap, a very unique overlap of actin and myosin, an optimal overlap to generate the greatest tension. If you're too on either spectrum, right, if you have no overlap or complete overlap, you're just not going to be able to create tension. All right, so that was the end of the changes in fiber length. So now let's talk about force generated by the whole muscle. So the whole muscle, right, if you want to generate different kinds of force, there's two things that come into play here: recruitment of motor units and then the size of the motor unit. So with recruitment of motor units. So let's just review real quick what a motor unit is. So a motor unit is a motor neuron. Okay, so this is our motor neuron. The body of the motor neuron lives in the spinal cord. Okay, and just to add a little bit of anatomy, right? This is our ventral horn of our spinal cord. So the body of our motor neuron is going to exit the axon, not the body. The axon is going to exit the spinal cord. It's going to come to your muscle and innervate a series of fibers. So you can see in this picture that there are some reddish fibers that are innervated by motor unit X. So X is going to just innervate these reddish fibers. Okay, so those are going to respond to the motor neuron X. Then our other motor unit is our motor neuron Y and all of its fibers that it is innervating. So our motor unit is defined as the motor neuron and all the fibers it innervates. Okay. So it turns out that the more motor units you activate, of course, the more force you can generate in that muscle. So this is how your nervous system works with the loading forces. So if you're picking up something light, again, something like a pencil or your cell phone, you only need to recruit one motor unit. You only need to activate only a few fibers within your entire muscle. You're not going to use your entire strength of the muscle at maximum tension, right? So you're only going to recruit a few. Maybe only one motor unit is firing. But as soon as maybe you want to pick up something heavier that requires more motor units, so you recruit another motor unit to help you deal with the load. So that is recruitment. So you're going to activate motor neurons in a fashion where you kind of activate the lowest one first, the smallest one first, and then you can recruit more as you go, as you need. So let's take a look at the size of the motor unit. And so before we leave, right, so this, just looking at this graph, So this is if only motor unit X was firing, right? So we have 5 fibers. This is going to generate a little bit of tension, right? This is like picking up your cell phone. If you want to do the motor unit Y is a little bigger. It has 7 fibers, so it can generate a little bit more tension, okay? But if you wanted to pick up a little bit of a larger object, say your physiology textbook, which is heavy, you're going to recruit both motor units so you can generate more tension because you're firing 12 fibers. Okay, so the size of the motor unit is how many fibers are innervated by a motor neuron. Increasing in tension occurs in steps proportional to the size of the motor unit. And this is kind of looking at this picture down here, the size of the motor unit. This is a small motor unit because there's only 5 fibers involved. This is a larger motor unit, unit Y, 7 fibers. Okay, so when you say large or small motor units, it just means how many fibers that one motor neuron is innervating. In this picture, this is called the size principle. Let me just back up by looking at letter B, the size of the motor unit. So you increase the tension proportional to the size of your motor unit. So if you have small muscles that are controlling very small, precise movements, that muscle will have very small motor units. So muscles of your eye that move your eye around, those motor units are tiny. There's 1 neuron for 1 muscle fiber. So what that does is it gives you very, very precise motion. You can also have really large motor units. Muscles for strength will have larger motor units. So if you think about like your calf muscle, your latissimus dorsi, you're going to innervate thousands of muscle cells per motor neuron because those muscles don't need to be precise. They just need to be strong. And then letter C, the fiber diameter. These are going to vary in motor units. The small diameter of the fiber is going to give you weaker movements, and the larger diameter of the fiber will give you stronger movements. So let's look at this one now. We kind of talked about letter D as well, motor unit sizes, right? The size of the motor unit and the fiber diameter are related. So if you have small motor units, we have small fibers, small motor neuron cell bodies, and small axon diameter. So everything's kind of small, generates not that much tension. For the large motor units have larger fibers, large motor neurons and cell bodies, and they have large axon diameters. It just takes more oomph, more action potential to get those larger fibers to fire. So you have larger motor neurons, larger axons to do that. Now let's look at the size principle. The size principle says that the order of the motor unit recruitment is related to the size of the motor units, and you recruit small to large. And that should make sense because you don't want to go all out and use like way too much force in a movement. It'll be very awkward if you start off with like way too much force and then just kind of backed down. You start off with a little bit of force, see that's enough, and then you add more motor units as you need. So your smaller units are recruited first. So you can see that here's our brain. We have our motor neuron here. So this is the pyramidal cell. Sorry, this is not a motor neuron. So this is coming from our brain. But then here, motor neuron X, motor neuron Y, motor neuron Z. So we can see there's 3 different motor units here, X, Y, and Z. And X will fire first. So it's the smallest run, right? So this is the smallest motor unit. And you can see that in the graph, if you excite X, then you get a small amount of tension. If you pull in, you need more tension, then you are going to activate that middle motor unit just to see if that's enough, right? So you're going to get more force with Y. And then it's going to be additive, right? And then if it's still not enough force, you're going to recruit the largest motor unit, and that's going to give you a big boost in tension. So you basically just go small to large according to what your body needs. Yeah, so basically for the size principle, it's the size of the motor unit. You just move from small to large. Larger neurons are more difficult to depolarize to threshold and requires greater synaptic input. So basically what happens is if you have a lightweight, your motor neurons, your brain says, okay, we don't need that many action potentials. Remember, because action potentials can sum and can give you greater tension. But once you start adding weight, the action potential levels will go up. And then you'll start to recruit your larger motor units, because those larger neurons need more input to create threshold. So with a heavier weight, that's going to create that greater input. And so those larger motor units will only fire when they're required, when you have those more active potentials coming in. All right. And then just real quick, before we end this portion, velocity of shortening. So this is basically velocity is speed, right? How quickly are you going to contract your muscles? And we're talking about isotonic, so we're actually moving the joint. So let's just do the example of like bending your elbow. So if you have nothing in your hand, right, you have zero load, then your maximum velocity is going to be you're at maximum velocity. You can do it as fast as you want, right? The fastest you can bend your elbow, because you have absolutely no load in it. Once you start adding weights to your hand and then you're trying to bend your elbow, what happens to the speed, right? So we can see the speed, the velocity is on this y-axis, it's going to go down. As you increase the load, right? So let's pick a spot here. What's a pretty heavy load? Pick a spot here, right? So as you increase the load, so let's take a look at this number 1. What's happening is that you've increased the load to an intermediate position here, and you just can't shorten as quickly, right? If you increase the load, what happens to the speed? It goes way, way down, because if things are too heavy for you to lift up, it's not as fast as if there's nothing in your hand. And that should just make intuitive sense to you. And then there's going to be a place where the load is greater than your maximum tension that you can create, or it's equal to, so you actually just can't move that object. You can hold on to it, but you can't move it. So your shortening velocity is 0. You just can't move it at all. So that's the curve that you see this is called the load velocity curve. Makes intuitive sense, right? As your load increases, the velocity is going to decrease. All right, some questions before we end this part. What is the long fibrous molecule that extends over actin monomers and blocks the myosin binding sites of muscles at rest? The answer is tropomyosin. Which 2 sites of the myosin heads on the thick filament are used to generate a muscle's mechanical force? Okay, so on the myosin head we have actin binding site and ATPase site. When does myosin have high affinity for the actin monomer on the thin filament? So high affinity, it wants to bind to actin. It's going to be in its energized form. What does calcium do to help the muscle contract? So calcium is going to bind to troponin, exposing the myosin binding sites. Because a muscle may contain hundreds of motor units, muscular tension can be varied by changing which motor units are active, activating the fibers within them. What is the name for the process of activating additional fibers of the muscle? Answer is recruitment. All right, so that's the end of part 1.